Thank you, Ben. Thank you all for coming. I'm really excited to be here and to talk to you about the intellectual history of coral reefs. There's really something about coral reefs that I think all of us find really compelling. That if it's the turquoise water or the white coral sand, all the fish and animals that live in a community on coral reefs. You can kind of see this picture here. This is of the Chagos archipelago in the Indian Ocean, which is near the Maldives, sort of off the western coast of India. And you can see the colorful corals, and there's fish that sort of look like corals right here. So I'm from Florida originally, and I also grew up in Singapore, which is a small island nation off the southern tip of the Malaysian peninsula. And we would often go snorkeling in the out islands out and around, and that's when I fell in love with coral reefs. And I think probably you're all aware of the problems facing coral reefs right now. Global warming. So scientists project a 1.5 degrees centigrade or even a 2 degrees centigrade increase in the temperature of the oceans. Ocean acidification is also a problem. As the ocean absorbs more carbon, it becomes slightly more acidic. And for limestone secreting creatures, that's bad because it makes it harder for them to make their shells, like coral. And just a few weeks ago, at the end of September, a report came out from the Intergovernmental Panel on Climate Change that was about oceans and the cryosphere, which is the frozen parts of the world, that the way the newspaper articles talked about it, and I just have a few examples here, the New York Times and CBS News, they really talked about coral reefs as facing death. Coral reefs are going to die because of these warming temperatures and increasing acidification, and also the language was very much coral reefs will face die-offs, coral reefs are dying. So when people are talking about coral reefs as dying, that means we think that they're alive. And it's not just the coral polyps that people talk about as dying, but it's the whole ecosystem that we're concerned about. And that means that we know that they're alive, and that is the modern concept of reefs as a single ecological unit, as a living unit together, functioning together. So how did we get there? So we're going to move back about 150 years, and you might recognize this guy. This is Charles Darwin. His first book was about coral reefs and how they formed before he wrote on the origin of species. So during the 19th century, especially the early 19th century, coral reefs were talked about in a geological sense and not a biological sense. And part of the reason why was people didn't quite know how they were formed. They didn't quite understand how such a small little animal, a coral polyp, could build this great big structure. And so Darwin's first book is about how these structures were formed. And his main theory was that so based on the idea that volcanoes in the ocean slowly sink beneath the surface, and as they slowly sink down, corals grow in a ring shape around the edge of the volcano. And as it slowly sinks down, the coral keeps growing on the top, and the base of the coral continues to go deeper and deeper and deeper underwater, where it can't actually grow because it's too deep. And then eventually the top of the volcano will be eroded or be underwater, and you get a lagoon and a ring-shaped island. And this was a very controversial theory. He also talked about how these ring-shaped corals would become fringing, so those are fringing reefs, and the development of barrier reefs, and the variety of types of reefs like that, but all in a geological sense. There was also the fear that during this time, shipping was the main way of getting around, and there was a fear that coral reefs grew so fast, they would create this block of coral around the equator, preventing ships from passing from the northern hemisphere to the southern and vice versa. So it was scary. The coral reefs were seen as scary and threatening, also because of shipwrecks, where you could lose your ship or your life or all the capital that a ship had. So this is what's happening in the 19th century. And this Darwin's theory became known as the coral reef controversy, and it was one of the biggest controversies, other than evolution, in the 19th century. And it wasn't solved until the 1950s. So this is an example of an atoll. So those are a group of coral islands in a ring shape. This is Eniwetok Atoll, which is out in the Pacific. If you came last night, there was actually another picture of one of the nuclear detonations done at this coral atoll by the US Atomic Energy Commission. In the late 1940s and 50s, they conducted a number of nuclear tests because it's very far out in the middle of the ocean. And so at the same time as they were doing this, they said, well, let's finally see if Darwin was right. And they bored down into the rock more than 4,000 feet deep, and they finally hit volcanic rock. So they proved that Darwin's theory was actually right, and that volcanoes sinking down slowly and coral building up is how many coral islands were formed. At the same time, they thought, well, let's study the ecology too. So two ecologists, the Odum brothers, H.T. Odum and Eugene Odum, conducted a study of the ecology of the reef in conjunction with the radioactivity. This is how they measured a number of chemical tracers that went through the reef. And it's called the trophic structure of a windward atoll. And trophic structure being the sort of food chain with a sense of how much biomass is at each level. So for example, the base of a pyramid of a food chain would be your plants. Those are your primary producers. And then the consumers eat the plants, primary consumers. Secondary consumers eat the fish that eat the plants, and so forth. And it was replete with assumptions of coral reefs as being ecologically rich, having rich life, being associations of plants and animals together. So that paper was published in 1955, and by that point, the modern idea of the reef is firmly in place, and it's an assumption in their paper. So my work looks at how did we get here when everyone else is talking about the geology of how reefs form? How did we start thinking about reefs as a unit? And the people whose work was essential for that was a group of botanists. And it's surprising to think about botanists and reefs, right? Because we think about coral and the animal communities that live on coral reefs. But I'm going to tell you how their work was really essential for developing the idea of reefs as living ecological units. So here we have some botanists. Top left, I'll point. This is Anna Weber-van Bosse, a Dutch algologist. And an algologist is someone who studies algae. Currently, if you talk to someone who's a scientist who studies algae, they would call themselves a phycologist. The only difference is that phycos is from the Greek word for algae or seaweed, and alga is from the Latin word. So, and seaweed is an algae. It's just not all algae are seaweeds, but some algae can be unicellular. Seaweeds are necessarily multicellular, and they also necessarily live in the sea. Underneath this picture is the Natural History Museum of London. That's where Ethel Barton Gepp, a British algologist, worked. I don't have any pictures of her. This is fairly common for women in science. Their archives are very small, and maybe they left the scientific field mid-career, and so they didn't quite get as high as they might have if they continued working. So that's where she worked, and that is a stand-in for her. And then I have two American algologists. This is Marshall Avery Howe, who worked at the New York Botanical Garden. And here is William Albert Setchell, who was an algologist and botany professor at the University of California, Berkeley. So why botanists? Why do botanists have something to say about reefs? And for the reason that botanists were thinking differently about how things lived on reefs is their insight was based on the idea that plants being sessile, being stuck where they're planted. They can't move, so they're much more dependent on the environmental factors around them for their livelihood. So things like how much light they're getting, the sort of substrate or soil they're on, if they're underwater plants, the depth. Those are all really important to the life or the aliveness of the plant, and therefore botanists are continually keeping in mind that plants are living beings. So I'm going to tell you more about Anna Weber-van Bosse, and she is best known currently as being the first female scientist to be officially included as a scientist on an oceanographic expedition. And that's the Saboga expedition, which I'll also talk more about in a second. These are some pictures of her, older, younger, a portrait in her lab. She was born to a very wealthy family. Her dad owned a shipping insurance company in the Netherlands that essentially had a monopoly on all the Dutch ships that were conducting trade with the East Indies, with the Dutch East Indies. And so she was afforded a number of opportunities that not everybody had, especially not all women at the time. Her Swiss governess encouraged her love of the natural world by taking her to the artist's zoo frequently, where she developed her love of plants and animals. But she was married twice, and even through her first marriage, she was taking classes at the University of Amsterdam. And she studied with Hugo de Vries, who is a very well-known Dutch botanist, and he's best known for his work on mutation theory and also as being one of the 3 rediscoverers of Gregor Mendel's pea plant experiments and their significance for the laws of inheritance and genetics. So, very famous, well-known botanist at the University of Amsterdam. She was one of 3 female students at the time, one of whom was related to de Vries and also of the upper class. And they were allowed to attend the lectures, but they couldn't go into the lecture hall until the professor was there. And they were allowed to do their lab work, but they had to do it in a separate lab room than the male students. At the University of Amsterdam, so she was taking courses there 1880 to 1883, she meets Max Wilhelm Karl Weber, who's a Dutch-German zoologist. He was hired to be a lecturer in comparative anatomy, and they get married in 1883. It was a very happy marriage in terms of their personal lives and their success in science. They would do collecting trips together. They went to Norway, where he would collect whale skeletons and also fish, and she would collect algae. They went to the Dutch East Indies on an overland trip in 1889 and 1990. He was collecting freshwater fish, and she was collecting algae. They went to South Africa, again collecting, and then importantly, the biggest expedition in both of their lives was to the East Indies on the Saboga expedition. Throughout this, is that still good? Okay. She is writing and thinking about the interrelationships between algae and some of the species that she finds it on. So her second paper is actually about algae that lives on sloth fur. A parasitic algae turns them green. And one of her later papers in 1890 is about the symbiosis between algae and sponges. And symbiosis becomes a common theme in her work and her life. She wrote some biographical notes on Max Weber for the Englishman who wrote his obituary, and she wrote of their marriage that symbiosis was such a good motto for their happy union. So in 1895, they moved from Amsterdam to Eerbeek, which is a town about 100 kilometers northeast of Amsterdam. And they bought a large estate where they had a number of pets, dogs, and a cassowary at one point. And that's where they both had lab space and workspace, and she kept her growing herbaria herbarium there. She also purchased others' herbarium collections, so she ended up amassing more than 50,000 specimens. And in a herbarium, you could have herbarium sheets that are, you have a dried plant, in her case algae, sort of slapped onto a nice sheet so you see the shape of it. And then she also had specimens stored in vials and preserving alcohol. So she had this great big collection, and this, along with their spacious estate, allowed them to host many visitors, and they became known as very generous and gracious hosts. And all of the other botanists that I mentioned, Gepp and Howe and Setchell, all visit her a number of times. And this picture here, this is a giant sequoia, was a gift in the form of a seed from Setchell, who was in Berkeley. So they're growing a giant sequoia in the Netherlands. So the Saboga expedition. This expedition is a Dutch expedition, 1899. They left March 7th, came back the end of February in 1900. And its main purpose was to measure the deep sea basins in the area. So this map shows you all of Southeast Asia, and they were focusing on archipelagic Southeast Asia, so from the southern tip of the Philippines and then around what is now modern-day Indonesia. It was the Dutch response to the Challenger expedition, which you might have heard of because it's one of the most famous oceanographic expeditions. It was a British expedition in the 1870s that was specifically studying the depths of the ocean and what lived there. And they did a number of soundings in and around the Dutch East Indies, and the Dutch thought that they should probably know more about their colonial territory, or know the most about their colonial territory. So in addition to the Saboga's goals of studying the deep sea basins, they were also studying the flora and fauna of the deep and shallow seas. And it was the first, it became the most extensive study of coral reef ecosystems up until this point. So this is the Saboga. It's a retrofitted gunboat. These are the ship's officers and the captain. So you can see this is Anna Weber-van Bosse here. Again, this is the first time a female scientist is officially included on the scientific staff, and she's in charge of studying the marine flora, or the algae. Max Weber is to her left. Tiedemann up here in the Navy peacoat is the captain. And then the other first officers are wearing the same naval outfit. And then there's 2 zoologists and a doctor also included on the scientific staff. This is the map from the Saboga monographs that shows their track. And I have a better map that you can probably see where they went and with the dates. So they started over here from Java, Surabaya. And they went all around, and this area now, so this tiny little bit of the Philippines up here, they did hit some Philippine islands, but this area now is what we call the Coral Triangle, and it's known as having the highest amount of marine diversity and also the highest concentration of reefs. And so the triangle is from the southern tip of the Philippines to the farthest east and farthest west bits of Indonesia. So they're doing extensive studies of this very rich ecosystem for the first time. So extensively. And what they're doing, here's the ship's whole crew. Anna Weber is here in the middle with a cat. And there's Max, and then the sailors and the whole staff. In order to collect the marine flora and fauna, they had a number of scientific instruments. This shows you, so they had dredge, sounding machines to record the depths. They had deep water thermometers to record the temperatures. Weber-van Bosse brought her microscope, although it proved difficult to look at under a microscope when the ship was moving, and also vertical nets, which you see here. Oops. This is another picture of the Saboga from a side view, and the red box shows you where the laboratory is in the boat. This is not the Saboga, this image, but I thought it was a nice pictorial view of the sort of tools that ships like these would use. So you can see the vertical nets, you could set it for different depths, a sounding lead, which would hit the bottom so you could know how deep it was, dredge, which digs up stuff from the bottom, and trawls. They also had plankton nets. So those are examples of what their tools would look like underwater. This is a close-up of the laboratory. So you see Max Weber here, and in front of him are these sort of aluminum tins. When they would pull stuff up from the water, they would dump it all out on a sifting table and then quickly sort of categorize it and throw different things in different buckets. So you'd have crabs in one bucket, squid in another, and fish in another, and then they'd bring it to the lab where they'd more carefully and selectively see what things they wanted to send back to Holland, and they would send them back in a preserving alcohol sealed up in these tins, and then they would end up at the Zoological Museum of Amsterdam, where they would later ask experts in that field of crabs or whatever to analyze all the specimens that they collected. And unfortunately, so when sea creatures die, they often lose a lot of the color that makes them so beautiful underwater. And also things like formaldehyde and preserving alcohols also cause them to lose their color. So they included an illustrator on their expedition, and this is J.W. Hausmans. He's the illustrator. So as they were categorizing stuff before they put it into the alcohol, he would do these beautiful watercolors of some of the invertebrates and fish to capture what they looked like when they were alive. They also, starting with this launch picture, this was their launch that was on the Saboga, a steam launch, and they would use it to get to shore or shallower reefs, and they're using casting nets. And then you can see one of the scientific officers had a camera, which they used to take some of these pictures. And then they also purchased specimens from locals. So here they're looking at what this diver, who's with the white beard kneeling down, brought in from what he had been diving for. So they relied on local knowledge as well. This is Hausmans, the illustrator, with a sailfish that he caught. And this is one of the few, it's almost 3 meters long. This is one of the few specimens that didn't make it back to Holland. They were running low on their preserving alcohols, and it's such a big skin, they decided to just put it in a sort of pickling solution, mostly salt water, and it was rotten by the time it got to Holland. But they did have a nice picture. And these are some examples of some of the illustrations by Hausmans. These are all sea cucumbers, and the images that you're looking at are from the Saboga monographs. So after the specimens were sent to Holland and categorized by experts, they published the results in this series of the Saboga monographs. And that's what's here at the Linda Hall Library. There are 147 installments of these publications, and held within 39 volumes. And we can look at some later, but I wanted to show you these ones because of the illustrations, and also because of this colorful sea cucumber here. It's red, white, and blue, which is the same colors as the Dutch flag. So if you've ever held a sea cucumber, you can find them on the beach or near a coral reef, and they just kind of lay there. They're kind of funny, simple invertebrates. And if you pick it up out of the water, its defense mechanism is to squirt water at you. And if it's very scared about your capturing it, it might regurgitate its entire gut out when it really doesn't want to be eaten. So that is relevant, I think, because when they found this, the zoologists were really excited. And they thought, oh, this is great. We can honor our queen and we'll name it after her. And then after thinking about it a little bit longer, they decided that it might not be such an honor to have a sea cucumber named after you. But these illustrations are by Housemans, and I think they're beautiful. So some more examples of some of the monographs and the different types of illustrations. There was starfish, chitons, which are like the armadillos of the marine world. They have this sort of armor, and these are more watercolors. You find them stuck on rocks right at the low tide line. And then we have the fish monograph, which Max Weber was an ichthyologist, so he wrote up the fish monograph. You might recognize some of these species. A triggerfish is a common reef fish, some sort of pufferfish. And the image on the right that looks like tusks, those are tusk shells or dentalium shells. And I included this one because the author is Mia Boisseven, who is another female scientist. She is a Dutch malacologist, someone who studies mollusks, and she wrote up this monograph. I mentioned that there were 147 installments. Of those, 22 were authored, either first author or second author, by women. So that's almost a sixth, which is pretty high for this time period. Most of them are published between 1901 and 1930. So there's quite a large number of female scientists included in this publishing effort, which the Webers were in charge of. Okay, so back to Anna Weber-van Bosse and algae. What you're looking at here is coralline algae or calcareous algae. And I think it's surprising because especially these two pictures here, it looks so much like how we think coral looks, especially when it's out of the water. So Anna Weber-van Bosse wasn't the first to say, hey, there's algae that produces a stony skeleton just like coral, but she was one of the first trained algologists to really focus on and study the role of this algae on a reef and in a reef ecosystem. What she found was that on almost every station that they collected at, there was lithothamnia, which is the type of algae that this is. It's a type of red algae. So if these were alive and in color, they would be a sort of pinkish or lilac color. And there's two types. So there's encrusting lithothamnia, which is this image here, and encrusting meaning how it grows. And it sort of grows how you might think algae would grow, like a lichen, like flat and over top of a surface. And that's important for reef structures because it cements the whole thing together and helps it persist despite erosive forces like waves and currents. So it's the encrusting lithothamnia is important for reef building, which her work sort of provides the basis for, and we'll talk more about who's making those points in a second. And then this is the branching lithothamnia that provides homes or places to hide for little reef creatures, just like coral skeletons do. So, like I said, she wasn't the first one to say, hey, these algae produce stony skeletons like coral, but she was the first botanist to do so. And what she was really important for doing is talking about the ecological conditions that they grew in, and also how prevalent they were across all their study stations. Many of the places that were termed coral banks were actually lithothamnian ridges or algal ridges. So she's proving the prevalence of these algae in terms of where and how frequently they occur. These are pictures included in the monographs of some lithothamnian reefs at low tide. Another calcareous algae that I want to mention briefly is Halimeda. So Ethel Barton Gepp was the British algologist, wrote up the monograph on Halimeda, and it is maybe more plant-like looking than lithothamnia. And you can see here, these are the illustrations from the monograph. And these sort of branching nodes have a calcareous hard structure. In the color picture, they're the green tufts here. And this is a deep sea reef in the Philippines, in the pictures from more recent, not from the Smoke expedition. But Halimeda is important because in some of the other attempts to drill down to prove if Darwin was right, they found that Halimeda structures, or calcium carbonate from Halimeda, made up the most mass of coral rock. So it's significant in how much Halimeda there is and how much it builds or contributes to coral rock. It's also, when it breaks down and erodes, it's what produces the white sand that we associate with coral reefs. So from there, those two monographs, Ethel Barton Gepp's and Anna Weber-van Bosse's, provide a large amount of data about where Halimeda and Lithothymnia are found, how frequently they occur in reef systems, and what sort of ecological factors allow them to grow. And from that, Setchell and Howe, the two American algologists, take it a step further and start arguing. Let's see, Howe's article is in 1912, that plants are the most important builders of coral reefs, the most important lime-secreting organisms that produce the coral reefs. So Howe says that in 1912, and then Setchell starts saying the same thing, and he starts going back to using the term nullipore, which is a zoological term that is not specific in terms of, it doesn't matter if it's Halimeda or Lithothymnia, and it was Lamarck is the one who came up with this term, but it's not specific in terms of, it doesn't refer to anything, sort of like plankton is not specific. It's just talking about a general type of thing. And Setchell continues saying plants are the most important reef builder, and he also starts talking about the reef builders and Lithothymnia and nullipores as symbiotic and the symbiotic driver of reefs. And eventually, in the 1920s, he starts defining reefs, as symbiotic entities. I'm not going to read these to you, but I want to point out that 1926, in a resolution by the International Coral Reef Committee, which Setchell is on, they define reefs as symbiotic entities. And in 1928, in a paper in Science, Setchell says they're symbiotic entities, they're ecological units, and they are treated, he treats them as one single aggregate. So by this point in time, the modern idea of the ecological reef concept is in place, and it's really based on these four botanists. So to quickly conclude, Anna Weber-van Bosse's life and her scientific work focused on the theme of symbiosis, and her work plus Barton Gepp's work on algae, on marine algae, where they found it, and what it did in reef structures was really important for providing the base of knowledge for thinking about reefs as symbiotic living entities. And Setchell and Howe took their evidence, pushed it further, and Setchell eventually defines reefs as symbiotic units. And I think I should make sure I say symbiosis is the idea that two or more organisms are living together to their mutual benefit, and that if they were apart, they would not be able to survive necessarily. And so lastly, why does this matter? So there's two reasons that I think this story is important, and one of them is about the future of coral reefs. And I think how we define a problem and how we draw a frame around it is really important for how we think of the solution. So thinking about how plants contribute to coral reefs might change how we think about solving the issue of coral reef death. A couple of reasons for that are, among corals and plants, they have different rates of limestone secretion, and they also have different mixes of the calcium carbonate and calcium magnesium carbonate that make up the stony skeletons, and those might have different reactions to an increasingly acidic ocean. And the role of the encrusting nullipore, I think, should certainly be taken into consideration in terms of trying to regrow reefs because of its role in structuring and cementing the reef together. The second reason is to tell the stories of women in science, because how we talk about the past influences how we imagine the future. And I think now there are many women studying science, and particularly biology, but the problem of the leaky pipe, where women are slowly lost at each successively higher level, drop out of sciences, I think could be part of the idea that how we view scientists as very professional, and we don't think about their personal lives. So the story of them having a very happy, successful marriage, I think, is an important story to add to the history of science. And thank you so much for coming. There are books, some of the rare books in the back that we could look at after, and I'll hand it over to Eric.
And we have time for a few questions. Mark's telling me to move over to get in the view of the camera. So raise your hand and I'll come by with the microphone. For those of you watching via live stream, just type in your questions and I will read them.
Yes, in your introduction, you mentioned the threats to the coral, and environmental was very key, but I'm curious about the pollution aspects, in particular plastics.
Oh, plastics. That's a great question. I think you're right. There are many more problems than just global warming and ocean acidification, including pollution from sunscreen with some of the chemicals in sunscreen. Overfishing is a big problem, and habitat destruction. I think we don't know yet how plastics are affecting coral, since it seems like they're coming up in all sorts of insides of things that live in the ocean as the plastic gets smaller and smaller and consumed. That's a good question. I'm sure it's not good.
And before we take the next question, I also want to reiterate what Emily mentioned, that at the conclusion of the program, you're welcome to join us across the hall to view some of the library works that Emily has been researching with while she's been here. Just please no food or drink in our History of Science reading room, but otherwise, you're welcome to join us.
Are there collections and their archives and these original drawings and watercolors all been preserved, archived somewhere?
There are, so the watercolors and a lot of the Saboga monograph collections are held in Holland at the city archives. It's actually interesting because they used to be held by the Zoological Museum archives, which is in the zoo. It's a building in the zoo. But because of the Netherlands low-lying landscape, the foundations of the building were rotten. And so they moved them to the city archives, which is open to the public. So even though it's not categorized yet, but they're there. Just hard to get to.
Emily, great talk, and on many different levels, very important topics, so thank you. I have a question related to any of the diaries that the women kept. This was very much focused on their science and the scientific writings that they did, but have you had an opportunity to explore any of their personal diaries?
That's a great question. One of my major sources is the correspondence between a number of these women. And Anna Weber-van Bosse kept all the letters and correspondence that she had with Howe, Setchell, Ethel Barton Gepp. There are two other female algologists who aren't published in the Saboga monograph, so I didn't talk about them here. But they formed a sort of network where they helped each other and they met each other. And two of them worked out of Roscoff, which is in the Brittany region of France, that sort of has a similar climate to Maine and tons of seaweed. So their letters, they're really excited about algae and doing all the sort of drudgery of naming and classifying it.
Any other questions? Yes. Got two more. I'll go over here and then come to you, sir.
What are some ways to fix coral reefs?
That's a great question. I think that the report that I mentioned talks a lot about what we can do to slow down our methane gas releases and sort of put a cap on the carbon monoxide, dioxide that we're releasing into the atmosphere. And actually, algae, potentially a different kind of algae, could save the day here. So the ocean is a carbon sink, and it's in most of the ocean. Algae is in most of the oceans, but it's not in the Southern Ocean, which is just around Antarctica. And so there's an idea that we could sort of make algae, the sort of unicellular algae that's a phytoplankton, if we could make algae grow in the Southern Ocean, it would absorb a lot more carbon dioxide and sort of pull it out of both the warmer parts of the ocean and also reduce our carbon monoxide levels. So that's one idea. That's bioengineering. And I think that what can we do personally? I think it's important to stay hopeful. And I hope maybe thinking about algae and plants and their role in helping reefs grow could provide more solutions. I think reducing our own carbon footprints, reducing plastic use could be potential ways. There are a lot of coral scientists doing good work on trying to help corals survive these warming events.
Yes, I have a question about our use of plastics. We produce enough plastic to kill every living organism in the ocean. I know there's a lot being done to improve that. Is that having an effect on production of coral reefs?
That's a good question. I don't think we quite know how plastic production is like a quantitative effect. I don't think we know quite yet. I do think that they are, well, another fun fact about algae that has to do with plastic. This is from the book Slime, which the library also holds. It's a 2018 book about algae. You can make plastic out of algae, and it's compostable, so it wouldn't cause same problems as more persistent plastics. Some of the athletic shoes create foam, like the cushion foam for their shoes, out of algae. So I think algae could save the day there too, but plastic-wise in the ocean, I don't think we know yet, and I think even the currents that bring plastics and trash to different parts of the ocean are still not quite fully understood.
Well, Emily, thank you for a wonderful presentation.
Thank you.